Okay, let's unpack this. Imagine a drug delivery system that doesn't just get absorbed and, flow through your body,
Uh-huh.
but instead it intelligently sticks exactly where it's needed
Right.
for precisely the right amount of time. Sounds almost like something out of science fiction, right?
It kind of does. But what's truly fascinating here is how often the most innovative medical solutions are inspired by, well, natural biological processes.
Ah, okay.
And today, we're taking a deep dive into that very concept. We're exploring the surprisingly sticky world of mucoadhesive polymers.
So our mission in this deep dive is to take your source, that's the polymers and mucoadhesive drug delivery systems, a brief note article, and really give you a shortcut to understanding the science here.
Yeah, the cutting-edge stuff behind these targeted, long-lasting drug treatments.
Exactly. You'll gain valuable insights into how these materials are revolutionizing medicine, making drug deliveries smarter and, well, vastly more effective.
Definitely.
So to truly appreciate this innovation, we probably need to lay some groundwork first.
Good idea.
What exactly are we talking about when we say bioadhesion, and maybe more specifically, mucoadhesion?
Okay, so bioadhesion at its core is simply the process where a material could be natural, could be synthetic sticks to a biological surface.
Like a glue for the body?
Sort of, yeah. Think of it like a specialized glue for living tissue. Now, mucoadhesion is a very specific type of bioadhesion.
Oh.
It's when that sticking happens specifically to a mucosal layer. That's that slippery protective lining we have in places like our mouth, stomach, nose.
Ah, right.
We've actually been using basic bioadhesive systems for a long time. Things like, denture adhesives or even some surgical glues.
Huh, that's really interesting. So it's not totally new then.
No, not at all.
I'm curious, when did scientists first really start exploring this idea of stickiness for, like, targeted drug delivery?
It actually goes back surprisingly far. The pharmaceutical journey with these mucoadhesive polymers, it dates back to 1947.
Wow, really? 1947?
Yeah, back then, researchers were trying to figure out how to make a penicillin drug stick to the oral lining
Yeah.
inside the mouth to treat infections.
Okay.
They were experimenting with natural stuff first, like plant gums, dental powders.
Mm-hmm.
Later, they found better results with some early cellulose-based materials. And this early work eventually led to commercial products like Adhesive.
Oh, I think I've heard of that.
Yeah, it was a blend designed specifically to adhere in the mouth. So it just shows how long we've been trying to solve this basic problem, keeping drugs where we want them.
Okay, so if you're designing a drug delivery system that needs to stick really well, what are the absolute must-have qualities for that polymer? What makes it, ideal for this kind of sticky situation?
Right, an ideal mucoadhesive polymer isn't just sticky. It has to be smart sticky.
Smart sticky, I like that.
Yeah, the core insight is that these materials are engineered to do several things at once. First, they need to adhere rapidly, right? Stick quickly, but without changing their own physical properties or messing up how the drug is released. That's key.
Okay.
Second, they absolutely must be biodegradable. They need to break down safely in the body without leaving any toxic byproducts behind.
Crucial.
Very. They should also be able to sort of protect the drug from enzymes at the delivery site. Enzymes that might break the drug down too quickly.
Ah, like a little shield.
Exactly. And finally, if the goal is for the drug to be absorbed into the bloodstream, the polymer should actually help enhance its passage into the tissue, not block it.
So it's doing a lot of jobs at once.
It really is. And to understand how these polymers stick, we first need to visualize that mucosal layer they're interacting with.
Okay, the mucus itself.
Right. It's mostly made of mucus, which is this fascinating gel-like fluid secreted by specialized cells called goblet cells.
Goblet cells, right.
And imagine very long, tangled spaghetti strands,
Yeah.
but made of special proteins and sugars called mucins,
Okay.
all suspended in, get this, over 99% water.
Wow, 99%.
Yep. It's these tangled mucin glycoproteins, along with various weak molecular attractions. Think of them like tiny fleeting magnetic forces that give mucus its hydrated, gel-like, and wonderfully sticky texture.
Okay, so we've got this fascinating sticky mucus layer. How exactly does the sticking happen on a, molecular level? Are there different ways these polymers latch on?
Yeah, absolutely. Scientists have proposed about 6 main theories to explain mucoadhesion. We can broadly group them into, say, chemical methods and physical methods.
Okay.
But the key takeaway, the gist of it, is that the process generally happens in 2 main stages.
Few stages.
First, there's the contact stage. That's just where the polymer and the mucous membrane actually get wet and make contact. Pretty straightforward.
Right.
Then comes the consolidation stage, and that's where the real physicochemical interactions kick in, forming a strong bond between the polymer and the mucus.
Ah, okay, so the consolidation is where the real magic happens.
That's where the sticking power comes from. And if we dive a bit deeper into those specific interactions, well, bioadhesion can happen through various molecular forces.
Like what?
You could have hydrogen bonds. Think of them as tiny molecular magnets forming between slightly positive and negative parts of atoms on the polymer and the mucin.
Okay, hydrogen bonds.
Then there are van der Waals forces. These are even more subtle, like really fleeting attractions between molecules.
Yeah.
Imagine incredibly weak, temporary clinginess.
Weaker ones.
Yeah. Then hydrophobic bonds. These happen when nonpolar parts of molecules try to escape water and cluster together, kind of like how oil and water separate.
Right, I get that.
You also have ionic bonds. These are stronger electrostatic attractions, like proper magnets, between oppositely charged molecules. This can lead to almost instant sticking.
Ah, okay, stronger again.
And in some cases, you can even form covalent bonds, where atoms actually share electrons. That creates a super strong, almost permanent stick.
Wow, okay, that's a huge range of forces involved.
It is.
So diving deeper into this, what allows scientists to actually control this stickiness? What specific factors can they tweak or manipulate to make a polymer more or less mucoadhesive?
That's where the real engineering comes in. It's a precise science of tailoring the polymer to interact just right with that mucosal layer.
How do they do that?
Well, first off, consider molecular mass, basically how big the polymer molecule is.
Okay.
Generally speaking, bigger polymers tend to be more mucoadhesive. Makes sense, right? More surface area, more points of contact.
Yeah.
So, for example, a polymer like polyethylene glycol or PEG might be barely sticky at a small size, like 20 kilodalton,
Right.
but it becomes much better at 200 kilodalton and shows excellent adhesion at 400 kilodalton. Some polyoxyethylenes up at 7,000 kilodalton are incredibly mucoadhesive.
Huge difference.
Massive. Then there's polymer chain length and flexibility. Think of it like a long, flexible rope versus a short, stiff one.
Okay.
The longer and more flexible the polymer chain, the better it can kind of weave its way into and entangle with that tangled mucin layer.
Ah, penetrates better.
Exactly. If you make the polymer too rigid, say by adding lots of cross links between the chains, it loses that flexibility. It can't swell as much with water either, and its sticking power generally goes down.
So flexibility is key.
It really is.
Yeah.
A clever strategy is sometimes to attach long, flexible chains onto a more rigid polymer core, kind of getting the best of both worlds.
Interesting. What else?
Functional groups are also absolutely crucial. These are specific groups of atoms sitting on the polymer chain.
Like hooks or something.
Sort of. Groups like hydroxyl, OH, carboxyl-OH, and amino-NH2 groups are fantastic for forming those hydrogen bonds we talked about.
Ah, the molecular magnet.
Precisely. Think of polymers like vinyl alcohol, acrylic derivatives, celluloses, starch. They're rich in these groups.
Okay.
And even more impactful are charged functional groups. These make the polymers what we call polyelectrolytes.
So electrolyte.
Yeah. Anionic polymers, the ones with negative charges like polyacrylic acid or carboxymethylcellulose, often stick very strongly because mucus generally has negative charges too, but they interact via other mechanisms like hydrogen bonding.
Okay.
And cationic polymers like chitosan, which has positive charges, can interact powerfully through electrostatic attraction with the negatively charged bits of mucin.
Yeah.
Plus, chitosan is known for being quite biocompatible.
So charge makes a big difference.
A huge difference.
Yeah.
Also, polymer concentration plays a significant role.
How much you use.
Exactly. There's often a sweet spot, an optimal range, typically around 1 to 2.5 weight%.
Not too much, not too little.
Right. Because if the concentration gets too high for some polymers, like polyvinyl pyrrolidone or polyvinyl alcohol, the polymer chains can actually coil up on themselves too tightly. This limits their ability to reach out and interpenetrate with the mucus.
So more isn't always better.
Exactly. It can actually reduce their stickiness, counterintuitively. And finally, you have to consider environmental factors. The pH, for instance, the acidity or alkalinity of the environment, can change whether those functional groups are charged or not.
Affecting the ionic interactions.
Precisely. Olson, for example, really shows its best mucoadhesion in neutral or alkaline conditions.
No.
Even the contact time matters. Initially, the longer the polymer is in contact with the mucus, the more time it has to hydrate, swell, and let those chains interpenetrate.
Makes sense.
And of course, just basic physiological factors like the natural texture and thickness of your own mucosal layer in that specific part of the body can influence how well any delivery system adheres. It varies.
That's fascinating how many different variables they can actually tweak. It's like tuning an instrument.
It really is a fine-tuning process.
So once we've engineered these, ingenious sticky polymers,
Yeah.
how do scientists actually put them to the test? How do they measure how well they stick?
Right. You need ways to quantify that stickiness. Researchers use a variety of clever methods, both in vitro in the lab and ex vivo, using animal tissues.
Okay, like what?
Well, one common approach is tensile strength measurement.
Tensile strength.
Yeah. Imagine you have two surfaces, one coated with polymer, the other maybe a filter paper soaked in mucin, or even a piece of excised mucosal tissue.
Uh-huh.
You bring them together, let them bond for a set time, and then you measure the maximum force needed to pull them apart.
Ah, like a tiny tug of war.
Exactly.
Yeah.
The higher the force needed to separate them, the stickier the polymer is deemed to be.
Simple enough.
Another method is the wash-off test. This tries to simulate what happens in the body more directly.
How does that work?
They typically attach a piece of mucosal tissue with the drug delivery system placed on it onto a glass slide. Then they put this whole setup into a standard lab apparatus like a USP tablet disintegrator, which gently agitates it in the solution.
Okay.
And they basically just measure the time it takes for the delivery system to completely detach or wash off from the tissue. It gives an idea of residence time.
So how long it would actually stay put?
Pretty much, yeah. For more advanced real-time measurements, there are specialized systems like BioCore integrated chip systems.
That sounds high-tech.
It is. It uses surface plasmon resonance, or SPR.
Mm-hmm.
Essentially, you immobilize the polymer onto a sensor chip surface.
Okay.
Then you flow a solution containing mucin over that chip. The system can detect, very precisely and in real time, when mucin molecules bind to the polymer.
Wow.
It gives you a very detailed picture of the binding kinetics, how fast it binds, how strongly it binds.
Very cool.
And then, of course, the ultimate test is often in vivo experiments conducted in living subjects, usually animals first.
Right, the real-world test.
Exactly. You might administer a delivery system that's been labeled with a radioactive tracer, for example.
Uh-huh.
Then you can track where that radioactivity goes in the body and measure how much is present in the target tissues at different time points. It directly tells you how long and where the system is staying.
It gives you the definitive answer, I suppose.
It's usually the most relevant data before moving towards human trials, yes.
So with all this talk of stickiness and testing, where are we actually seeing these cutting-edge systems making a real difference in the body? What parts of the body are proving to be like the best spots for this kind of targeted delivery?
That's a great question. Mucoadhesive systems are incredibly versatile, and they're being applied in several key areas where traditional drug delivery often faces challenges.
Like where?
The oral cavity, specifically the inside of the cheek, the buccal mucosa, is a prime example.
Okay, the cheek lining.
Right. It's super easy to access, obviously.
Yeah.
And a huge advantage is that drugs absorbed there go directly into the bloodstream.
Ah, bypassing the liver.
Exactly. They avoid that first-pass metabolism in the liver, where many drugs get broken down before they even have a chance to work. That's a massive benefit.
Yeah, definitely.
Plus, the cheek lining is relatively immobile compared to, say, under the tongue, which makes it really suitable for controlled, prolonged release using sticky patches or tablets. Polymers like cyanoacrylates, polyacrylic acid, Gitosin are used here.
Okay, mouth makes sense. Where else?
The nasal cavity is another important site. It offers rapid absorption for some drugs.
Right, nasal sprays.
Exactly. But the nose has this built-in cleaning mechanism, the mucociliary clearance, that usually removes things pretty quickly, like within 15-30 minutes.
Ah, washes it away.
Yeah. So mucoadhesive polymers are vital here because they can significantly extend the time the drug stays in contact with that nasal lining, giving it more time to be absorbed. Think HPMC, carbopol here.
Got it. Improves the window for absorption.
Precisely. Then there's the ocular cavity, the eye.
Eye drops, yeah. Always feel like they run out immediately.
They often do. Tears and blinking constantly wash away traditional eye drops, leading to really poor drug absorption, very low bioavailability. So mucoadhesive ocular inserts or patches can combat this. They stick to the eye's surface, keeping the drug there much longer and significantly improving its effectiveness. Thiolated polymers, polymers, are examples here.
Makes a big difference for eye treatments.
Huge difference. Both the vaginal and rectal lumens are also important sites. They can be used for both local treatments right there in that area and also for systemic absorption into the bloodstream. Again, bypassing the liver.
And another way around the liver issue.
Right, and mucoadhesive polymers here help prevent the suppository or gel or whatever form it is from migrating away from the target area. Keeps the drug where it needs to be. Polycarbophil is common here.
Ensures it stays put.
Exactly. And finally, of course, the gastrointestinal tract, the stomach and intestines, has been a major focus for a long, long time.
Yeah, seems obvious for pills.
It is. There's always interest in controlling how long a drug delivery system stays in specific parts of the GI tract, like making it stick to the stomach lining or reside longer in the small intestine where most absorption happens.
To improve absorption.
To improve absorption, or maybe deliver a drug locally to treat something like an ulcer. Chitosan, polyacrylic acid, alginate are often explored for GI applications.
That really highlights the versatility, doesn't it? Sticking drugs in all sorts of places.
It's incredibly versatile.
So let's get into the specifics then. What are some of the, the star players in this sticky world? What are the different classes of polymers that actually make these systems work?
Okay, yeah, let's explore some of the main categories. First, we have the broad class of hydrophilic polymers.
Hydrophilic, water-loving, right?
Exactly. These are polymers that love water. They swell up and can even dissolve in aqueous environments like body fluids.
Okay.
Now, the key insight here, as we touched on, is that charged hydrophilic polymers, the polyelectrolytes, are generally much stickier than neutral ones.
Right, the charged ones.
Anionic ones with negative charges, like polyacrylic acid and carboxymethylcellulose, CMC, are super common. They form strong hydrogen bonds with mucin. And then the cationic ones, like chitosan, with its positive charge, interact powerfully through those electrostatic forces with a negatively charged mucin. And they're usually very biocompatible, too.
Good combination.
Yeah. We also use neutral hydrophilic polymers like poloxamer or HPMC, methylcellulose. They might not be as inherently sticky, but they're often used to, say, make liquid formulations thicker or design tiny drug-filled microcapsules.
So they have other roles, too.
Definitely. Next up are hydrogels.
Hydrogels.
Imagine these as three-dimensional networks of polymer chains, all cross-linked together. They can soak up and hold a tremendous amount of water, like a super absorbent sponge.
Right.
Now, generally, if you make a hydrogel too tightly cross-linked, too rigid, it actually reduces its stickiness. Because it can't swell as much, the chains aren't as flexible. They can't, penetrate with the mucin as effectively.
Ah, that flexibility thing again.
Exactly. Although, there are exceptions. Sometimes, clever design using specific cross-linking can actually increase adhesion by packing more sticky functional groups into a smaller volume.
Okay, so it's nuanced.
It is. Hydrogels are particularly exciting for delivering larger molecules, like peptides or proteins, and they've shown real promise in improving how well poorly water-soluble drugs get absorbed.
Interesting applications.
Yeah. Then we have the thiolated polymers, or thiomers.
Thiolated. Sounds specific.
It is. These are polymers that have been chemically modified to have free thiol groups, have a H sticking off them. Think of them as molecular hooks.
Hooks, okay.
These thiol groups are designed to form very strong covalent disulfide bonds, SS bonds, with cysteine-rich parts of the mucin proteins.
Covalent bonds, the really strong ones.
The really strong ones. This creates a much stronger, more permanent adhesion compared to just hydrogen bonds or van der Waals forces.
Wow.
And this enhanced stickiness can also do something interesting. It could boost what's called paracellular uptake.
Paracellular, meaning between cells.
Exactly. It helps the drug squeeze through the tight junctions between the mucosal cells rather than having to go through them, which can be difficult for some drugs. Thiolated polyacrylic acid and thiolated chitosan are leading examples here.
So, stronger sticking and better absorption pathway?
Potentially, yes. Yeah. It's a very promising area.
Cool. What's the last category?
Finally, there are lectin-based polymers.
Lectins. Those are proteins, right?
They are. Lectins are proteins often found in plants, but also animals and microbes, that have this unique ability to bind very specifically and reversibly to certain sugar or carbohydrate molecules on cell surfaces.
Like a lock and key.
Exactly like a lock and key. So these polymers use lectins as targeting agents. The idea is to make the delivery system stick only to cells that have the specific sugar keyhole that the lectin key fits into.
Highly targeted.
Very targeted. Now, there can be some concerns about potential toxicity or immunogenicity, body reacting against them with some lectins.
Ah, okay.
But researchers are exploring specific ones, like wheat germ agglutinin, WGA. It seems to have lower immune reactions, and it binds well to the lining of the intestines and the lungs,
Making it good for
making it potentially very useful for targeted oral or perhaps even inhaled drug delivery systems.
fascinating stuff. So, wrapping this up then, what does this all really mean for medicine for patients? It seems like scientists are finding these incredibly clever ways to make drugs work smarter, not just harder, by quite literally making them stick to their targets.
Absolutely. If we connect this to the bigger picture, it's about fundamentally optimizing how drugs interact with our bodies.
Hmm.
We're moving beyond just hoping enough drug gets absorbed.
Right.
It's about using material science and understanding biology to vastly improve a drug's residence time, how long it stays where it needs to be, and its bioavailability, how much actually gets absorbed effectively at very precise locations.
And the result?
The result, hopefully, is more effective treatments, needing maybe lower doses, and potentially fewer side effects for you, the patient.
It truly is about tailoring these delivery systems for much better outcomes. And it shows us that sometimes, the simplest sounding idea, just making things sticky, can actually have the most profound impact when you apply sophisticated science to it.
It really can. And maybe a final thought to leave you with. Consider how this level of targeted adhesion could genuinely transform treatments for chronic conditions. Imagine reducing the need for frequent dosing injections, multiple pills a day, and how that could drastically improve patient compliance, just making it easier to stick to a treatment plan.
Yeah, that would be huge.
So the question is, what other areas of medicine, what other conditions could really benefit from therapies that just, well, stick around longer where they're needed most?
Yeah.